Elrigu Namaskara. Today I'll be dealing on the topic, the type study Marchantia genus, its reproduction and its sporophyte. Well, I'm Dr. Jalakshmi, Assistant Professor of Botany at Postgraduate Department of Botany, Narani Science College, Pullman, Mysore. So this is a topic of bryophytes in first sem BSc Botany in the syllabus of University of Mysore. Coming to the objectives of what is dealt in this session is the study of the reproductive stages of Marchantia, the types of reproduction, that is vegetative, asexual and sexual methods, the study of the sporophyte and the alternation of generation in the life cycle of Marchantia. Coming to the vegetative reproduction, there are different modes of vegetative reproduction which is found in bryophytes. In Marchantia, we find these type of reproduction, that is fragmentation, then adventitious branches and gemma. Gemma, of course, is also considered as a type of asexual reproduction as it is considered as a propagating organ. And coming to sexual reproduction, it is oogamous. All the species of Marchantia are dioecious, while male sex organs are produced on are the antheridia, and the female sex organs are called archegonia, which is produced on the male thallus and the female thallus. Coming to the first type of vegetative reproduction, it's a progressive death and decay. That is, mature cells are present at the posterior end and which are usually short-lived. These cells, new cells are regularly formed by the apical notch. So death and decay occurs naturally, therefore, from the posterior end, and as the process of decay starts reaches the point of dichotomy, the two apical parts of the thallus will separate. So in a mature thallus, from the posterior end of it, the decaying starts of the thallus, and each branch of the dichotomy will separate out and give rise to new plant. Coming to another type of vegetative reproduction, it is by adventitious branches. Some species of Marchantia produce adventitious branches on the ventral surface. So this is a ventral surface. From the ventral surface arises small appendaceous-like branches, which is called as adventitious branches, which on separation will develop into a new thalli. Well, another major type of reproduction which is found in Marchantia is by the production of gemma. The extensive distribution of Marchantia is mainly because of the production of this propagating structure that is called as gemma. Gemma cups are found on the dorsal surface of the thallus along the midrib region. So this is the gemma cup which is produced on the dorsal surface of the thallus. So usually the margin is hyaline. You find a hyaline colorless margin which is low, spiny or entire from the floor of the gemma. So here within this what we find dark green structures are the gemma, and this is a gemma cup on the surface of the thallus. When we see this in a vertical section, we can see this is the thallus which bears the rhizoids, and this is the assimilatory filaments. And on the thallus, you find this cup-like structure which is called a gemma cup. And this within this gemma cup, many small stalked discoid biconvex gemma arise from the floor of the gemma cup. Gemma is mainly constricted in the middle. We find so this is a single gemma. We find a constriction in the middle. It's called as a notch, which possesses two apical cells. So on either sides of this constriction or the notch, we find two apical cells. Hence, the gemma can give one single gemma can give rise to new plants. The cells gemma contains chloroplast in most of all the cells, while some cells are lacking the chloroplast, which are called as rhizoidal cells, and some of the cells are filled with oil. Some mucilage hairs also arise from the gemma cup floor. So within this gemma cup floor, along with the gemma, we find some mucilage hairs which imbibe water and help in a dispersal of the gemma when it is mature. These mucilage cells imbibe water, and by the friction of the mucilage hairs and the gemma is really is detached from the stalk, and it falls on a suitable substratum and gives rise to a new thallus. Gemma, on coming in contact with ground, it starts germinating immediately and gives rise to a thallus. Each gemma can give rise to a two different thalli. So this was the vegetative reproduction of Marchantia thallus. Coming now to sexual reproduction, Marchantia is heterothallic or dioecious plant. The male thallus bears the anthridiophores and female thallus bears the archegoniophores. They arise from the distal end of the thallus from the growing point, and after their formation, the growth of the thallus will cease. You will find them at the tip of the dichotomy where the apical notch is situated, and that is utilized for the formation of this specialized gametophores. Hence, the formation of the thallus will stop once the gametophores arises from the apical notch. Coming to the male reproductive part, that is the anthridiophore, which is usually 1 to 3 centimeter long stalk and bears a 8-lobed peltate disc. So, if we see it as such, this is a 8-lobed peltate disc of the anthridiophore with a long stalk of 1 to 3 centimeter. Internally, it's similar to the thallus of with an upper epidermis, air chambers, photosynthetic filaments, then the storage region, and there is a rhizoidal groove. The antheridial chambers on this peltate disc usually alternate with air chambers. You find the antheridial chambers alternating with the air chambers, which open out with an air pore, and within the air chambers you find the photosynthetic filament. Each antheridial chamber consists of a single antheridium, opens externally by a pore which is called as the ostiole. Each lobe of antheridia arises acropetally. You find first formed or mature antheridia in the center and newly formed antheridia towards the apex, that is, they are arranged in an acropetal succession. Antheridiophore has two longitudinal grooves where we find the rhizoids and the scales. Coming to a mature antheridium, which is globular or oval with a multicellular stalk. So this is an oval within antheridial chamber. There is a single antheridium with a multicellular stalk. The body of the antheridium has a single layered jacket enclosing androcytes which will metamorphosize into antherozoids. The antherozoids, the minute biflagellate structure. The mature antheridium will dehisce, that is, the water enters in through the ostiole. Some cells of the distal end will disintegrate when they come in contact with water, and the antheridium will rupture, and the androcyte mother cells will metamorphosize into antherozoids, and from the ostiole or the opening, a cluster of or a group of these antherozoids will emerge out like a smoke column and spread on the surface of the antheridial disc. Coming to the female reproductive structure, it's the archegoniophore. Again, it's a modified gametophore which bears the archegonium. It has a slightly longer stalk, about 2 to 5 centimeter, and the terminal disc, which is 8-lobe structure. Again, this is an 8-lobe structure. Archegonia are born on the dorsal surface. You find initially the archegonia on the dorsal surface of the thallus in an acropetal succession. So you find the archegonia that is on the dorsal surface in acropetal succession. Internally, if you see the TS of vertical section of the archegoniophore, we find a upper epidermis having air pores, then photosynthetic filament, then the storage region. Once the one single antheridia gets fertilized, then there is the curvature of the lobes or the rays of the archegoniophore, bringing the archegonia towards the ventral surface or towards the lower side of the surface of the archegoniophore peltate disc. A mature archegonium, if we see, is a flask-shaped structure which consists of a short few-celled stalk and a globular venter which bears an egg and venter canal cell, and the neck bears about 4 to 5 neck canal cells, and the apical part is covered by 4 cover cells which open out when the egg archegonia is mature. The swollen venter usually consists of a single layered jacket enclosing ventral canal cell and a large egg. So when the archegonia is mature, the cover cells will set apart and keep the archegonia open for the entry of the antherozoids. So for this process starts with the fertilization. That is, water is very essential for fertilization. Archegonia is placed upright on the archegonial disc. Ventral canal and neck canal cells will disintegrate and form a mucilaginous substance which imbibes water and results in the separation of the cover cells by pressure. Now the archegonia is open now for the entry of the antherozoites. The antherozoites present on the surface of the antheridial disc are splashed by raindrops onto the surface of the archegonial disc. They are attracted chemotactically towards the neck of the archegonium, and antherozoites enter archegonia, and one of them will fuse with the egg, leading to the formation of a diploid zygote, so, which ends with the process of fertilization. Now, each of this fertilized egg or the zygote, which is a post-fertilization product, will give rise to a sporophyte. First, it gives rise to an embryo, and then that gives rise to a sporophyte. Each sporophyte is a structure which is differentiated to a foot, seta, and capsule. It is enclosed by a protective covering called as calyptra, and also another protective covering called as perigynum, which encloses a single sporophyte, and a pericachium or involucre, which will uh, cover the group of sporangia. So the calyptra is a protective covering, that is the capsule wall is formed, that is the wall of the venter, while perigynum and pericachium are additional surrounding covering of the sporophyte. Coming to the different parts of a sporophyte, we find a foot which anchors the sporophyte and absorbs water and nutrients from the gametophyte. Seta is a connecting capsule structure connecting the capsule and the foot, which is a few layers in thickness and few layers in height. And coming to capsule, it is a single layered wall enclosing a mass of spores and elatas. Elatas are hygroscopic in this way, that is help in the spore dispersal. So initially in a mature sporophyte, it is filled with the spore mother cells. So spore mother cells undergo reduction, division, giving rise to haploid spores. Some of them will develop into elatas, and some of them will develop into spores. Elatas are hygroscopic and help in the spore dispersal. Sporophytic generation of the life cycle ends in the formation of haploid spores. These haploid spores are released from the sporophyte once the sporophyte is mature. The seta elongates, it pushes the sporophyte out of the perigynum and pericachium, and the capsule wall will rupture, releasing the spores. These spores are carried away by wind or rain and fall on a suitable substratum and gives rise to a new thallus. The spore is that's why the first cell of gametophytic generation. It is surrounded with a thick wall which is differentiated into outer thick exine and inner thin intine. Each spore mother cell of Marchantia produces 4 spores, out of which 2 of them will develop into a male thallus and 2 develop into a female thallus. So all these spores are haploid. Since Marchantia is dioecious, 2 of the spores give rise to a male and 2 of the spores give rise to a female thallus. So the cycle starts with the development of the thallus, so which gives rise to a gametophyte plant body. So in the life cycle of Marchantia, we find both the gametophytic and sporophytic generation, which alternates with each other, while gametophytic generation giving rise to the male and female reproductive structures, male and female gametes, which fuse by fertilization, giving rise to a zygote, which starts the sporophytic generation, and the zygote gives rise to a sporophyte, which will produce the haploid spores within the sporophyte. That haploid spores again will give rise to the gametophyte. So, gametophyte and sporophytic generation alternate in the life cycle of Marchantia. So, these are the references which has been referred to prepare this presentation. Thank you.